Hello and welcome. My name is Sebastian. I'm the director of CNS research at the company Cellectricon. Here at the company, I'm responsible for the neurodegeneration research and also related business area. Today, I would like to present to you our concept, how we aim to decipher the spreading of neuropathologies in neuronal circuits using our high-capacity microfluidics platform. So first, I will shortly talk about our company. So Cellectricon is a collaborative services provider. We are dedicated to accelerate drug discovery and also research in the area of chronic pain and neurodegeneration. Our team comprises of highly experienced neurobiologists, stem cell researchers and engineers, and we have a proven track record of developing innovative in vitro assays of enhanced translational value. So our company is located in Gothenburg in Sweden. We are a privately held company. We were founded in 2003 as a spin-out from the Chalmers University of Technology. And initially, we started as a product company, but then transferred to a service model in 2012 in order to align with the industry climate and the market needs. And today's presentation, I would like to start with describing neuropathologies seen in Alzheimer's and Parkinson's disease patient brain. So what you can see here in this image is the immunohistochemistry staining of cortex tissue isolated post-mortem from an Alzheimer's disease patient brain. And this tissue was preceded by a fixation and using a tau antibody in order to visualize tau tangles within the soma of the cortical neurons here pointed by the arrows. And also what you can see here, the arrowheads pointing to it are so-called tau tangles within the neurites. So this is a typical pathological feature one can observe in brain tissue isolated from Alzheimer's disease patient brain. And similar to this, one can also observe in Parkinson's disease patient brain tissue so-called alpha-synuclein inclusions within the soma here pointed by the arrows or the neurites here pointed by the arrowheads in substantia nigra neurons. So these are typical neuropathological features in these neurodegenerative diseases. But as you can see here, we have these neuropathology features in cortical neurons and substantia nigra neurons. However, in fact, it turns out it is not only in these brain areas where you can see these pathologies. In fact, that is shown in this slide now. Here you can see by the color codes in blue for tau tangles and for red, for alpha-synuclein inclusions in red. You can see that at early stages in the time course of the disease, indicated below here in years, that one can see these pathologies in subcortical regions, for instance, in the brainstem or in the medulla oblongata. So, and during the time course of the disease, apparently these tau tangles spread into, or alpha-synuclein inclusion spread into other brain areas. And at the end, and let's say at some kind of end stages of the disease, one can observe that nearly the entire brain show these neuropathologies, and therefore one is talking about progressive neuropathologies in Alzheimer's and Parkinson's disease. Of course, this aspect is highly interested to study and to reveal what exactly is happening here in the patient. And in the next slide, I will just describe you a little bit by using let's say, a schematic drawing, describing here a current hypothesis, which is used to explain this, let's say, progressive neuropathology seen in vivo. And as you might heard about, there's a concept, it's called the prion-like spreading of neuropathologies, which I would like to describe here. So initially, we have a healthy neuron, which for currently unknown reason shows at one point pathological hallmarks, for instance, accumulation of misfolded proteins. But also this affected neuron started to secrete these misfolded proteins. And an important aspect of the prion-like spreading hypothesis is that these misfolded proteins can be uptaken by axons, which are derived from neurons, innervating the primary affected neuron. So this is shown in this slide here, in this schematic drawing. And this then, once this misfolded protein is within the axon, it becomes axonal transported. So in that case, it's a retrograde transport leading to the somatic accumulation of this misfolded protein. But however, also what it does is it causes misfolding of endogenous protein in this recipient neuron. So therefore, we have started with like a primary affected neuron, and then this releases a misfolded protein, and which in the end leads to pathology seen in a secondary affected neuron that could be localized, let's say, in a far distant brain region. So at Cellectricon, we have decided to model this progressive neuropathology seen in the patients in vitro by using and devising a high-capacity microfluidics platform. So our aim was here to be able to do high-content analysis of pathological processes in compartmentalized neuronal cultures. But also we had to aim to devise a system which has sufficient capacity for screening in the search of compounds that modulate specific cellular processes that are involved in progressive neuropathologies. What we have came up with is shown here in that slide. So we developed a 384 well plate, so it looks like a regular 384 well plate. However, this 384 well plate comprised of 96 of this here shown experimental units. So what you can see here is a 3-well system. So we have a well 1, well 2, and well 3, and these wells are interconnected by microfluidic channels. And we use those systems in order to culture primary, but also human iPS cell neurons in the system. And then we can also, as shown here in this in green, we can culture, for instance, iPS cell cortical neurons. Here, these cells were cultured for 3 weeks in vitro, and then they are filled with the calcium-5 dye. To one point is to assess the functional properties of those cells, of these neurons, but also to visualize the neuronal morphology and the neural growth within the channels. That is one application we have used, and that was part of our technical validation procedure, just to observe if we can culture primary and human iPS cell neurons in the system. Another part of our technical validation procedure was to assess if we can maintain fluidic integrity in the system. So what that means is that at one point we want to apply molecules in the well, let's say in well 1, and we want that this molecule, or let's say seed, is only affecting the cells in well 1, but does not diffuse into interconnected well 2. So in order to test it, we did a testing series where we used different molecular weight fluorescent molecules, for instance, fluorescein. So here shown in the schematic drawing, we added fluorescein into well 1, and then we created a hydrodynamic barrier by having exceeded volume in well 2. And by this, then, we create a fluidic integrity. Therefore, that means that molecule in well 1 does not diffuse into well 2. How we test it and how we measure that? For that, we assessed, we did imaging settings in well 2 to measure the fluorescence, here shown in this diffusion profile plot. And what you can see is actually that we do not measure any type of fluorescence when we're having a hydrodynamic barrier in well 2. In contrast, when we don't have different volumes in the system, then we can see that over time there's an increase in the relatively units of fluorescence detected in well 2, and that indicates that molecules has been diffused from well 1 into well 2. So what this experiment here shows is that we can maintain integrity in the system over more than 3 days without any fluidic crosstalk or leakage in the well. And this is, we think it's an important aspect one need to address when using those systems. So in the next few slides, I would like to show you a case study where we use that system for in vitro modeling of pathological processes occurring in Parkinson's disease. As I showed you before, Parkinson's disease is characterized by the spreading of alpha-synucleation within the patient brain. And on cellular level, we have several processes going on here. We have axonal uptake of so-called seeds or misfolded proteins. axonal transport, somatic accumulation, and misfolding of endogenous proteins. So all these aspects, cellular processes, we aim to model in our system. And indeed, in the next few slides, I will explain to you how we have succeeded here. So what you can see here is the description about the methods we're using in order to generate compartmentalized primary neuronal cultures. So first, we isolate embryos from C57 black 6 mice. These are E18 embryos, and then we perform tissue dissection of the cortex, and then generating a cell suspension from this cortical tissue, and then we culture cells in the outer wall of these experimental units. To simplify that here a little bit, and for the next few slides, I will just talk about the well A and well B. And as you can see here in the schematic drawing, we culture cells in well B, but the well A is without cells. And you will see later why we do this approach. Next, what we do is we taking fluorescently labeled alpha-synuclein. We take a specific form. It's the sonicated preformed fibrils because we know that those are seed competent, meaning they can cause misfolding of other proteins, for instance, monomeric alpha-synuclein. So we use now this fluorescent labeled seeds and apply this one to well A at 6 days in vitro. Then, of course, we maintain fluidic integrity in the system, so we create a hydrodynamic barrier by having different volumes in well A and well B, as I described before to you, and we obtain the material using for our experiment from Stressmark Biosciences, because this company also provide a very good characterization of this material. So we know from the electron microscopy imaging that we're having really fibrils, which we're applying here, but also the seed competence has been evaluated by theoflatin T assay. So therefore we know we're using seed competence material in our system. After 7 days of incubation with the seeds, we use an operator high content imaging system from PerkinElmer and also the related Harmony high content analysis software in order to characterize our model system, but also describe the cellular processes occurring in this disease model. So what you can see here is a characterization work we have done because we wanted to know what type of cells we have in culture to better understand our model system. So we culture about like 20,000 living cells per well. And then we have here performed immunocytochemistry staining using NuN as a marker for neurons or GFAP as a marker for astrocytes, which is shown here at this the image on the left side. And we also did the quantification of the neurons and astrocytes, and we observe we have like a 1-to-1 ratio, about like 40% neurons and 50% astrocytes. Also, we combine other markers to characterize more the neuronal identity, which we have here in the system. And for that purpose, here I show an example standing for PAC6 in neuron positive cells, and we estimate and quantify that about like 75% of the neurons we have in the system are PAC6 neurons, so indicating an excitatory phenotype of those cells. So these are most likely these are glutamatergic neurons we have in the system. So next here, what I want to show you in this slide is the compartmentalized cultures we are using for the system, and also that slide will explain to you why we're adding only cells in well B and leave the well A without cells. What you can see here is a staining conducted 7 days after in vitro, and you can see beta-tubulin positive axons which grow into the cell-free interconnected well, and these axons are generated by the neurons in well B, which we here visualize by map2AB, and this is a marker to visualize the somatic dendrites. So what you can see here is that we can really nicely generate compartmentalized neuronal cultures in the system, which then enables us to specifically access and monitor neuronal compartments. So we can access the axons, but we also can monitor and visualize the soma and dendrites, and thereby can expose the different part of a neuron to different, let's say, seeds in that case. So exactly that is what we have done here. So what we did, and I described it before, we used the alpha-synuclein seeds, which are fluorescently labeled. We applied this to the well A, so we applied this to the axonal net, 6 days in vitro. And as you can see here, when we wait 7 days, we can see really nicely that all the axons nearly are filled with the alpha-synuclein seeds, which are fluorescently labeled. And also then what one can observe is that at the same time, you see when you take an image in the interconnected well B, so basically where the neurons located, which send the axons into well A. So what you can see is this red or white dots, and each of these, let's say, dots in that sense represent cells who somehow taking up the seeds by the axons in well A. And therefore, this indicates that the axons contains the seeds, and the seeds become retrograde transported and appear in the cells interconnected or located in well B. Then we also confirmed that these cells are truly neurons. We applied here new N staining. So we performed immunostatic chemistry staining to confirm really that the cells having the seeds are neurons. And interesting what you can see at that time here, 7 days after application, that most of the neurons, or nearly every neuron who has a seed, is located at the entrance area of well B. But we do not see any cells at the far end of well B. So what that means is that the entrance area of well B is the area where the channels are present connecting to well A. So all the neurons, in fact, having the seeds are localized at that area, but not at the far end. Next, to understand more where are the, let's say, the neurons localized that project from well B into well A, we used here a dye, in that case dye I, and that we apply to the axonal net in well A. And as you can see here, this dye I is also nicely uptaken by the axons. And when we now take a look into the well B, again, we see here dots, and these dots represent, as I described before, dye I-filled cells, and also immunostatic chemistry confirmed that these dye I cells are UN positive neurons. So that shows really the position of neurons here in the system that project the axons from well B into well A. And as you can see here, you see most of the neurons which are, let's say, transwell projecting neurons, so they're projecting from well B into well A, are localized at the entrance area of well B. However, you see here like 3 individual neurons localized in the middle of the image field of well B, and those are also dye I-filled. So that shows that you just understand the dimension here. That shows that they in the system comprise neurons which are able to cross distance between 3 to 4 millimeters. So the system comprises very long projecting neurons. And I think that is important to understand also to describe the disease progression in our system. So next then, we combined alpha-synuclein seeds, so they are fluorescently labeled, with the dye I seed. So again, we applied both together. We did a co-application of that seeds and the dye I solution in well A, and we waited several times. This data is shown here after 7 days. And what you can see is here on the right side, the dye I-filled cells representing the interval connecting neurons. And on the left side, you can see alpha-synuclein seeds fluorescently labeled. What we observed is 2 populations of neurons, 2 populations of interval connected neurons. So one group of the dye I-filled neurons, they're having the seeds, and as you can see here below, here's an example of 4 neurons, and you can see 3 out of these 4 neurons showing the seeds in their somata. However, we see that most of the neurons, which are dye I-filled, do not have the seeds. So that means that these neurons, they project over to well A, so they have the axons over there, and these axons are also exposed to the seeds. However, those neurons do not show a somatic accumulation of the seeds, which somehow indicate that the neuronal culture we are using comprise subcortical neurons, which are, let's say, susceptible for seed uptake. In contrast, most of the other neurons in that system do not taking up the seeds. So this is a very interesting point we feel, and that's something we are currently further investigating. So because we want to understand which type of neurons are capable of taking up the seeds, or put it another way around, which type of neurons are able to, let's say, protect themselves not to taking up the seeds. So those questions we think are very interesting, and we are pursuing more work here to decipher this more in detail. However, a next question, of course, one can ask is, okay, what happens with the cell having the seeds inside the soma? And what you can see here now, what we did is, again, the same experimental design. However, what we did at the day here, we used an antibody which indicate phosphorylated endogenous alpha-synuclein. And then you can see here when we did an image in the red A, where we have the axonal net, you can see there's a lot of alpha-synuclein speckles, so indicating we have here some kind of the presence of a misfolded endogenous alpha-synuclein within the neurites. Then we wanted to know how does the neuron look like, and what you can see here is again immunocytochemistry staining, where we used an antibody for neurons, so in that case NeuN, just to visualize the neuron. And as you can see, this NeuN positive cell here contains the alpha-synuclein seeds, the fluorescently labeled seeds in red. However, also when we then apply the antibody to detect the phosphorylated endogenous alpha-synuclein, you can see really a strong, some kind of a Lewy body-like accumulation of this endogenous alpha-synuclein in that cell. So that shows that this in line with our concept, which I introduced before, that the seeds can be uptaken, by the axons transported, retrograde transport and accumulate in the soma, and there they cause pathological phenotypes, which are some kind similar to the in vivo situation. Interesting is that when we now take an overview look on this well B, we see, and that is shown here, this is an example image, you see here again, a little bit highlighted on the right side, the cell having this, let's say, Lewy body-like accumulation of endogenous alpha-synuclein, but we see only few cells having that at that time point. So after 7 days, we see only few cells having this particular phenotype of pathology. So what are the next steps? So shortly, what is the outlook of our activities here right now? We want to assess more in detail the neuron-to-neuron alpha-synuclein spreading and propagation process. So there are several parameters we are currently investigating. So one, of course, using different cell models. So as you might know, there are different transgenic mouse lines available. For instance, there are mouse lines available expressing human alpha-synuclein, having an overexpression of those proteins. It would be very interesting to see how these cellular models behave in our platform. That is something we are very interested in. Also, as you might know, seeding material, so meaning the protein like alpha-synuclein can have different post-translational modification, which has an impact on the seeding capacity. And then it would be also very interesting to see if that has an impact on the spreading and propagation capacity. Also, this aspect is very interesting for us, and that's something we're pursuing. Also, in this presentation, I showed you data about like a 14 days in vitro experimental paradigm. Of course, one can expand the experimental duration, so that's also something we're doing. And one can expand that to 4 weeks or 6 weeks in order to assess more, let's say, the temporal course of the neuron-to-neuron spreading of these misfolded proteins. However, we feel that our concept to use neuronal labeling or combine neuronal labeling with fluorescently labeled alpha-synuclein seeds, and also using imaging of misfolded protein, here in that case phosphorylated alpha-synuclein, will help us to better understand the process, what is happening in our system, and how one can really then decipher the neuron-to-neuron transfer of the seed material. So an important question we are asking ourselves and try to aim to answer is like, does a fluorescently labeled alpha-synuclein seed and endogenous protein aggregation occur in a neuron which is not DII labeled? So what that means, as I showed you before, we have some kind of a primary affected neuron. In that case, in our model system, it would be then the di labeled neuron. But if we would see a cell which is not di labeled, but this cell has fluorescent labeled seeds or misfolded protein, then we would see that this cell would represent a secondary affected neuron. And that, in our opinion, would represent a more specific approach to prove the prion-like spreading hypothesis more in detail in an in vitro system. So to summarize, what I have shown you today is that we have established a neuronal compartmentalization approach in using our high-capacity platform. By this, we are able to monitor cellular processes which are involved in progressive neuropathologies, such as the axonal uptake of alpha-synuclein. I showed you data about the axonal transport for alpha-synuclein and how the seeds then at the end accumulate within some of the cells. And also I showed you data that the seeds cause misfolding or evidence for misfolding of endogenous proteins. Here in that case, it was alpha-synuclein. And then, of course, what you can envision here is that this approach we're doing is not only applicable for alpha-synuclein, it's also applicable for other neuropathological proteins such as tau or TDP43. And that's also something we are highly interested in. However, we are also, just to click here, what we're currently evaluating in our alpha-synuclein model is transsynaptic spreading or propagation. So I talked about this before, our approach, how we want to do this. Also, we are highly interested in what happens with the neuron having the seeds. Does this individual neuron having an impaired neuronal function? We think that our optical electrophysiology capabilities are very suitable for that. So we can really decipher here driver excitability and synaptic communication, and is that impaired in that system? And also we are interested to see and observe axonal and neuronal degeneration, which is also a hallmark of these diseases. So our aim here is, by using this approach, is that we want to assay several disease mechanisms in one native neuronal model. So by that, we believe that our concept enables to address fundamental scientific questions, and we believe that our concept enables pharma clients to obtain molecular fingerprints of molecules, and therefore this is suitable for the decision-making along the drug discovery path. So I would like to thank you here for your attention, and feel free to contact us for any questions you have or inquiries you would like to discuss with us, and have a good day.